And that story says that you have within your own brain and spinal cords your own stem cells right now that are capable of inducing repair within the nervous system. So I think that's a very important point. It's one that we didn't know of maybe 5 or 6 or 7 years ago, that there exists within the brains and spinal cord of every one of us until the day we die, our own stem cells sitting there, waiting to potentially repair in the case of injury. Now, what's quite interesting is that in response to injury, they become activated and begin to divide and begin to migrate to the site of injury. Now, we initially thought that this happened only in birds. And the reason is it was identified first in birds who acquire new songs when they meet different mates. And it turned out that it was there in the birds, and it was required for the birds to learn new songs. And if you halted the division and differentiation of these stem cells into neurons in the bird brain, the bird started giving old songs to a new mate, and the new mate didn't buy it and went bye-bye. You see? Right? So that tells you a very important point, that making these new neurons within your brain is critical for at least the bird to learn new behaviors. So is that true in humans who are learning new songs? Well, the answer is absolutely yes, that they contribute to new behaviors, they contribute to mood, they contribute to a bunch of things on a normal basis. That's not even talking about injury. Okay, so that's a very important point. We'll see if we can show a few slides of that. Now, that's the good news. Okay. So, it's not even important that I show this. This is in a rat. This is how we identified that they were there. We now know in humans that they are there. So that's the good news. The other good news is that there are ways to modulate within your own brain what the stem cells do. And I'm going to show you that in a second. The bad news is that in response to injury, they kind of fail. So they try to repair a transverse myelitis lesion. They try to repair a stroke lesion, and they fail for reasons that we are now beginning to understand. So here are the things that alter what you do with your own stem cells in your own brain. One of the things, exercise, good. Okay? So you can put a rat on a treadmill, and the stem cells within that rat's brain will just start to divide and migrate and do all kinds of good things, right? Stress reduction increases the amount of stem cells and what they do. Sleep alterations, people waking up all the time, not getting enough sleep, decreases the number of new neurons or new neural stem cells. Depression decreases, which is why Adam says you got to treat the depression. And which is why Adam says if you treat the depression, you're not just treating the depression, you're treating the disease, the transverse myelitis or the ADEM or the multiple sclerosis. Why? Because what happens if you treat the depression? The neural stem cells start to divide again. So that's pretty cool. Anxiety decreases. And then there are some biological things that we're studying that you can't really control. Growth factors like BDNF, alteration in proteins called Notch and things like that. And then there are these undetermined signals that cause them to fail in injury that we're trying to now overcome. And we have some exciting data in terms of how to overcome this block. In other words, the stem cells get to the site of the injury, and then they kind of try to do some stuff, and then they kind of give up, and they just sit there and don't do anything helpful. If we can overcome that and allow them to finish the deed, you might be able to either protect the at-risk neurons or remyelinate demyelinated neurons. And so we have some new data as to how to do that, which is pretty cool. The conclusion then, harnessing the power of endogenous stem cells will be a powerful approach to the treatment of neurologic disorders, and you can start that right now, right? So that's, I think, a very important point. Now, when they fail, we have to think about exogenous stem cells. We have to think about transplantation. So I very briefly want to tell you about 2 vignettes as we are progressing toward getting them to clinical trials. One is a cell called a glial-restricted precursor. Those of you who heard me say this yesterday, I apologize for the redundancy, but I need to kind of explain to you. You've heard that the embryonic stem cell is really the mother of all stem cells. It can become anything, which is both good and bad. A glial-restricted precursor is farther down the differentiation pathway, and it cannot become everything. But that's good, because I don't want it to become everything. I want it to become 1 or 2 things. I want a stem cell, when transplanted, to become an oligodendrocyte to remyelinate or an astrocyte to secrete growth factors. And this cell is able to do both, but can't do anything else, which makes it less likely to cause problems with safety. So this is what we are studying in an animal model of transverse myelitis. And we've created an animal model of transverse myelitis, and we are transplanting these cells into the spinal cord of the animal with transverse myelitis. And we are doing these studies under FDA guidance because we're going to use these data to justify a clinical trial. This is what they do, and I won't bore you with all of this. I'll shift for fairness. Green here is myelin. So you've taken an animal that has no myelin and dies, and you transplant these cells in and ask if you get myelin, and the answer is very, very robustly yes. And the animals survive for a really long time when they would otherwise die. This is not my work. This is Steve Goldman's work, but it's really beautiful work, and he used the cells that we are using. When you look under a really, really high-power microscope, you've seen some electron micrograph pictures, you look at myelin. Now, this is a really good look at myelin. What you're seeing is the section, a corner of an axon. So that's the wire. And what you're looking at here are these wraps of myelin, right? Because that's what an oligodendrocyte does. It first puts 1 layer of insulation around, then it says that's not enough, keep going, keep going, keep going, keep going. And here you've got 20 wraps. And that's good. The more wraps you have, the more insulated the wire is, the better it conducts. So these cells, which are human cells, transplanted into that mouse, myelinate in a really beautiful way. Okay, our first indication is transverse myelitis. The first clinical trial will be in patients with transverse myelitis. This is being done in collaboration with a company called Q Therapeutics, for which I get nothing, just to make that clear. But in fact, I didn't even know they existed, but now that they do exist, they can kind of help get this to a clinical trial. They were going toward pediatric leukodystrophies, and we said, no, no, no. You got to do transverse myelitis. It's perfect for this, right? And they've agreed. They've shifted their whole company direction. I like that. Why transverse myelitis? This is the argument I made to them, but I know I'm preaching to the choir here. It's 1 lesion in 1 spot of the spinal cord, unlikely to come again, and the pathology of that lesion is 2 things. Demyelination, good, because I can make oligodendrocytes. And axonal injury, good, because you can make astrocytes which protect them from dying. More importantly, surgery would be quite easy in the spinal cord, and what we're thinking about is transplantation directly into that lesion. And we have these biomarkers which are key to establishing whether it's working. We have imaging that can determine whether or not it's working. We have electrical studies. Many of you have gotten evoked potentials, which can define if it's working. We've got quantitative vibratory clinical tests, which can help us determine if it's working. This is coming. Now, it's our planned IND. In other words, we're submitting all of our data, we hope, to the FDA in February of 2008. That's not too far away. And the FDA would then evaluate that and give us the rights to begin enrollment in a clinical trial sometime in 2008. Now, who's going to be the first people to get this? People who are fairly recent in their transverse myelitis, not people who are years out. Not yet. The first thing we have to do here is win. We have to get something that works. So you've got to pick the patient population in whom that is most likely to occur, which is people who are closer to the injury. If that works, boy, things start to accelerate, right? You do proof of principle in the population most likely to work, and then you broaden it out. By the time that we broaden it out to patients who have had transverse myelitis for a longer period of time, we will know even more how to modulate, how to make a nice, good bed for these cells once they get into a chronic lesion, right? You just heard about what a chronic lesion looks like. Well, that's not very hospitable for the transplantation. So you have to kind of change the bed of the chronic lesion to make it accept the transplanted cells a bit more. That would follow, okay? So that's coming. Won't do that. Won't do that. Now, last vignette, a few minutes. Now we go to a different thing, and this is to make motor neurons. Not oligodendrocytes and astrocytes, but motor neurons. Motor neurons are the neurons that start in the spinal cord and reconnect with muscle. They come from embryonic stem cells, which is the mother of all stem cells. Why? Because no other stem cell can make a motor neuron. So adult stem cells and all of these other things you heard about are good for some things, but nothing else can make a motor neuron except for an embryonic stem cell. And the embryonic stem cell can make not millions, not tens of millions, but hundreds of millions of them. That's good. And I'll just tell you that the age of embryonic stem cell biology is really about 5 years old. The reason for that is, though we've known embryonic stem cells have been around for about 2 decades, we couldn't control what cells they made until 5 years ago. They would become cells of various cell types, but we couldn't figure out how to direct them to become one particular cell type. So when I'm transplanting into the spinal cord, I don't want cartilage cells in the spinal cord, right? All I want are motor neurons. So to be able to harness the potential of the embryonic stem cell is really kind of time zero for embryonic stem cell biology. We're now 5 years into that, and we can make motor neurons, and this is a beautiful set, 2 motor neurons. They're just beautiful. I don't know if you appreciate it, but that is derived from an embryonic stem cell, and by all criteria, this is a motor neuron. And we've then done a bunch of studies in a dish asking if they would reconnect with muscle in a dish. They do. And then we transplant them into this poor little thing, which is a rat which is paralyzed. And it's paralyzed. We did that. We had to create a model of this as well. But it's paralyzed because all of the motor neurons in that animal's spinal cord are gone, and therefore there's no connection to muscle. Therefore, the muscle doesn't move. So let's see what we did. This is a cartoon of what we did. If we could hit that, it should come up. And there's Ben Greenberg narrating this. I don't know if you'll be able to hear Ben Greenberg narrate it. All right, I'll be your narrator. All right, so this is the upper motor neuron. Uh-oh. And the analogy here is the house. There's 2 wires in this house. The light switch goes on, and the electrical current goes down to the second wire. And if the second wire is connected, the light goes on, and the room gets lit. Now, if that second wire is not there, the electrical impulse stops, the light does not go on. So now how about the spinal cord? You've got a 2-wire circuit, therefore the muscle moves. In paralysis, a lot of the causes of paralysis are that second wire is gone, therefore no light going on, therefore no muscle movement. So what we've done is tried to replace the second wire, which means you've got to get it into the spinal cord, but then you've got to get it to connect with muscle. So we put the cells into the spinal cord. And then we have to do a bunch of tricks to get them to become the motor neurons, and then 2 other factors, BDNF and NT3, to cause the surrounding environment to recognize those cells as friend and to form connections with them. More factors to get the axons out of the spinal cord, and yet 1 more to get it to connect with muscle. Now the light goes on. The muscle moves. And I'll show you what the rat does in real life other than that cartoon. So if we go to the next one now, you'll actually see a few rats. Yeah, so just back to the PowerPoint. That one, yeah. You can go full screen on that to make it just a little bigger. If you just right-click and then full screen. So this is one of the rats, and he can't move his hind limbs. And that's because all of the second wires are gone. This goes on for a little bit as he gets into his Mary Jane wrapper. So this is data now from several years ago, but we've got more recent data which came out last summer in that we could really reconnect, and the animals got significantly better because of that reconnection. All right, so here is one animal that has recovered, and it hasn't recovered that well, but it can now put its hind limbs under it. One limb you can see is stronger so that it circles around a little bit, but it now pushes off with both of its legs there. So that is one level of recovery, and here's the best recovery that we now see more frequently than the other level. This animal is obviously much better. And that recovery in the rat takes about 6 months and lasts for the life of the animal, which is about an additional 6 to 8 months. So they didn't get all the way better, but they got somewhat better. So if we can just now go back to the PowerPoint. So where are we with that? That's great to be able to do that in the rat. And it is a really important biological proof of principle that you can rewire a part of the nervous system. First time that's been done. So let's go to the next one. So where are we now? So we have to now do the same studies in anticipation of a clinical trial. Next slide, if you would. So you have to do large mammal studies under FDA guidance to justify doing this in humans. So we've created a model in a large mammal that would allow us to do this. We actually use something called ricine to induce a very slight weakness, and then we transplant into that animal. And we have to use non-federally approved human embryonic stem cell-derived motor neurons. And the reason we have to use non-federally approved cells is because the federally approved cells stink. And they don't do what we want them to do. They don't become motor neurons, and they're not genetically normal, and they don't behave in any way like you would want them to, and I wouldn't trust them going into a human patient. So that's not good, because that puts us in a little bit of a quandary where you have to use non-federally approved funds, and therefore you cannot use federal funding. But nonetheless, these studies are being done under the FDA doesn't care about that. They just want it done in a safe manner, and they want all the data gathered for safety, for toxicity, make sure there are no side effects in these animals, and then submit that data. And the proposed IND for this would be not 2001, that's a mistake, 2011. Why that long? Because these are more dangerous cells. And we have to really make sure that there are no side effects. We have to do studies within these animals that lasts for, a couple years. So that's going to take some time. Meanwhile, we are working through the patient populations. Again, the first patient population will be infants with a fatal genetic disorder called spinal muscular atrophy. So this is 100% fatal in infancy because of a genetic disorder that causes loss of motor neurons. And again, the first patient population has to be the one in which there would be the greatest likelihood of success, and then you expand out from there. And so there are lots of reasons why that would be the ideal choice. But then the expansion, obviously, would be into adults who would need to reconnect over longer distances, obviously because of size, and at progressively more remote periods after the acute injury. So that's all coming. And we are pretty certain that these trials are going to get underway. We don't know what they're going to show, but we're going to proceed forward. So that's where we are. That's my update. I think is there any more slides? Oh, those are my daughters.